Hi class. Today we are going to be talking about meiosis. Meiosis. We've already talked about mitosis. Now we're moving on to the process of making gametes. So first thing we need to do here is remind ourselves of a few terms. Obviously we need to know what genes are. Gene is a section of DNA that codes for one protein, and these are passed from parents to offspring. Our second bullet here says genes to proteins to traits. This is called the central dogma of molecular biology. Basically, genes encode proteins, and the specific combinations of proteins that an individual has determine many of their traits. Each of these genes is made up of a sequence of DNA nucleotides with the bases A, T, C, and G. And so one gene would be different from another gene based on the sequence of those bases. DNA replication, which we learned about, is how DNA is copied before a cell divides. Generally, it's copied exactly, though occasionally there are mistakes. But to go from parent to offspring, you need specialized cells called gametes. So these gametes are going to be the sex cells, sperm and egg cells generally. And so during fertilization, two gametes combine, bringing genes from both parents together. And this is why genes, you're going to have half as many chromosomes in a gamete as you will in a somatic cell. As you can see in the image for humans, we got 23 chromosomes in an egg, 23 chromosomes in a sperm, leading to 46 chromosomes in a zygote. So here we compare the two types of cells. Somatic cells, remember we said, are body cells, except for sperm and egg. So this is almost every cell in the body besides sperm and egg cells. In humans, we're going to have 46 chromosomes in these cells, two full sets, 23 from mom, 23 from dad. We call these diploid cells because they have two sets, di for diploid. And we abbreviate this as 2N, so two complete sets. Whereas gametes are going to be the sperm and egg cells. They are made, instead of being made in mitosis, they are made from meiosis, which we will talk about today. They contain only 23 chromosomes, half as many because they're going to combine. And they are called haploid. Haploid means one set of chromosomes, which we abbreviate N. So for humans, 23 chromosomes in a haploid cell, 46 in a diploid cell. And so comparing the two types of reproduction, we learned a little bit about asexual reproduction in mitosis. This produces genetically identical cells from a single parent. Very little variation, but you do get some variation because of mutations. It's faster, it's more energy efficient. Whereas sexual reproduction, you have meiosis producing gametes. Two parents combine genetic info to produce the offspring. One of the benefits of this is much more variation in diversity, which is very useful when populations are adapting to environmental changes. However, this type of reproduction is slower, takes more energy, and so that's a disadvantage compared to asexual reproduction. This is an example of an organism that can reproduce asexually, a hydra. This is budding. One can separate from sort of the parent, but it's a cloning process, so you don't get genetic variation, but it is faster and easier to reproduce this way. Binary fission is the method that bacteria use to reproduce. Also quite a bit simpler than sexual reproduction. They only have a single circular chromosome to copy before division, and then in binary fission they separate into two distinct cells, but they're going to be either genetically identical or very similar with only small changes due to changes in replication. And here we've got the life cycle of a plant, okay, which does involve sexual reproduction, getting the pollen from one plant to hit the stigma of another plant, which can then grow a pollen tube, to fertilize the egg, to make a seed, which can grow into a new plant. So most plants use sexual reproduction. And so in human cells, we can tell the chromosomes apart by their size, where the centromere is, and the banding patterns. So we can stain the chromosomes and then see different banding patterns, and this allows us to tell them apart. This kind of picture is called a karyotype, where we line up all the chromosomes in order with their homologous partner. So the 2 copies of chromosome 1 are lined up, the 2 copies of chromosome 2, and so on. Homologous chromosomes are chromosomes that are not identical, but they carry the same rough sequence of genes. So for example, chromosome 1 from mom and 1 from dad are very similar. However, they will have some different alleles on them. Mom might have a different allele at a certain gene than dad, but these are homologous because they are quite similar. They carry roughly the same sort of characteristics. In humans, pairs 1 through 22 are called autosomes, and the X and Y chromosomes are called the sex chromosomes. Autosomes basically just means every chromosome besides the sex chromosomes. And in humans, the way we traditionally define sex is having 2 X chromosomes is categorized as female, and having an X and Y chromosome is categorized as male. So before the karyotypes are organized, you will get the stained bands, but the chromosomes will not be in any sort of order. Once you organize them, you could get something like this. And so take a look at this picture, and you want to make sure you can look at this and first quickly be able to tell, is this karyotype from a male or female? So take a second to look at it there. You should have seen that there are 2 X chromosomes and no Y chromosome, which means this karyotype is from a female. Okay, another example here, similar, but now we have 1 X chromosome and 1 Y chromosome. You can see the Y chromosome is much smaller than the X. So this karyotype is from a male. This is a karyotype from breast cancer tissue. One thing we've seen in many cancers is abnormalities in chromosome number, extra chromosomes, pieces of extra chromosomes, which is rare in normally functioning cells. And so since we've learned about the cell cycle, it says predict what happened in cell cycle regulation for this to occur. One of the things is you can get non-disjunction, where the chromosomes do not separate properly. So instead of splitting 1 to each side, sometimes 2 go to one side, sometimes a piece of 1 goes to one side and the other piece goes to the other, instead of splitting evenly. All right, and so here's another image describing homologous chromosomes. Okay, so again, they're not identical. All right, if you look here, down here for this gene, these 2 chromosomes have different alleles, big C versus little c, but you can still see each gene is roughly in the same place on each chromosome, a similar order of genes. You might have identical alleles, as some of these are identical. And you get 1 of these chromosomes again from dad and 1 from mom. And we refer to these as homologous. It's very important to understand the distinction between homologous chromosomes and sister chromatids, which we explain right here. So we just talked about homologous chromosomes. Non-homologous chromosomes would be like chromosome 2 and chromosome 10, okay? Different genes, no real similarities, could be different sizes, centromeres into different places. Sister chromatids, on the other hand, which we've learned about before, are when 1 chromosome copies itself, okay, and they're still paired together. Those are identical. So same genes and alleles. This is after DNA replication, okay? And so sister chromatids, unlike homologous chromosomes, have the same origin, and they are literally identical, whereas homologous chromosomes, you got 1 from one parent and 1 from another, and they do not have to be identical at every locus. All right, so let's get into a bit about the process of meiosis. So we've learned a bit about the cell cycle, with interphase being the stages G1, S, and G2, okay, and you get DNA replication in stage S, okay. Meiosis, unlike mitosis, is two divisions. So you have meiosis 1, where you go through the 4 stages, PMAT, prophase, metaphase, anaphase, telophase. In meiosis 1, the homologous chromosomes separate, whereas in meiosis 2, the sister chromatids separate. And we'll go over that distinction a bit more. This produces 4 daughter cells that are haploid instead of 2 diploid daughter cells like mitosis did. So you get half the number of chromosomes, but you do have one full set. Like you have a copy of chromosome 1, a copy of chromosome 2, a copy of chromosome 3, and so on, but you only have one copy of each instead of 2. So here we see our cell. Okay, we have the sister chromatids are the duplicated copies of chromosomes. Okay, and then we can have homologous chromosomes with the same general sequence of genes. So in our first division in meiosis 1, we start with prophase 1. Every chromosome pairs up with its homologue. So homologous chromosomes pair up, in other words. So they find each other and they match up. Then a very important process occurs called crossing over, where they literally swap pieces of chromosomes. This increases the variation and can produce new versions of chromosomes not found in either parent. This crossing over location is called chiasmata, right? And you can see that marked here. But this is very essential because it increases genetic variation, even beyond the variation that would already be created by the random assortment of these chromosomes. You actually create new versions of the chromosomes. So if you look here, you can see crossing over can occur between these two homologous chromosomes, and you end up with a copy of a chromosome here and here, okay, or sorry, I should say here and here, okay, that were not present in either one of the original cells' parents. So we've had crossing over occur. So now in metaphase 1, the good thing about meiosis is once you've learned the stages of mitosis, there's not a lot of differences, so you just have to highlight sort of the important ones. The homologous chromosomes line up in the middle, okay? M metaphase, M middle, so they line up in the middle. Okay? In anaphase, the homologous chromosomes separate. So in mitosis, the sister chromatids separated. Right here, the homologous chromosomes separate. So later we can separate again. Okay, so right now we've separated the homologous chromosomes, but the chromosomes are still replicated. They still have their sister chromatids stuck together. In telophase 1 in cytokinesis, you get 2 haploid cells. Because you only have one set of chromosomes in each cell, even though it's replicated. That doesn't count as 2 different sets. And so each of these cells is already genetically different from the parent. And then, of course, you're going to get the membrane pinching in, forming the cleavage furrow, and separating in cytokinesis to form 2 new complete cells. One thing to keep in mind here is after meiosis 1, these cells are already haploid. Okay? A lot of times people think, well, you copy your DNA, so you have twice as much, then you divide, so you're at the normal number, and they think diploid, and then they divide again, haploid. No, because the way the chromosomes separate in meiosis 1, they've only given each of these cells one set of different chromosomes, because these sister chromatids do not count because they still are identical, except for crossing over. So after meiosis 1, these cells are already considered haploid. Okay, they just have one set of each chromosome, but it's copied. Okay, and that doesn't count as 2 different sets. So in meiosis 2, we're going to do a similar process, but now instead of separating the homologous chromosomes, we are going to separate the sister chromatids. Okay, we do not copy the DNA again, so there's no DNA synthesis between these 2 divisions. We're starting already with 2 haploid cells. Metaphase 2, these chromosomes line up in the middle. Okay, they're not identical now because crossing over has happened, but they're still considered sister chromatids. Anaphase 2, we separate sister chromatids. Okay, so I keep doing this hand motion because it is useful to think about. Okay, meiosis 1, homologous separate. Meiosis 2, sister chromatids separate. Okay, and so now the chromosomes are no longer replicated. Telophase 2, cytokinesis, we're going to end up with 4 haploid cells, each one with one set of chromosomes, no, not a replicated set now, and they're going to be all different from each other and from the parent. So where do we get all this variation from? One way is the independent assortment of chromosomes. The way the chromosomes line up, if you look at this picture here, okay, which ones are on the left, which ones are on the right, okay, is random. So it's not like all the chromosomes that the person got from dad are on the left and all the chromosomes from mom are on the right. No. So chromosome 1, the way it lines up, has no effect on chromosome 2 or 3 or 4. So you have this random assortment of chromosomes, which creates a huge number of possible combinations. So because you have 23 chromosomes and there's 2 options for each, that's 8.4 million combinations just based on independent assortment without even crossing over in there. Then another way to get variation is crossing over. Recombinant chromosomes, where they swap pieces. You tend to get 1 to 3 crossover events per homologous pair. Can be more, but that's generally on average. And this adds a huge amount of variation even on top of random assortment. And then another further source of variation is random fertilization. Okay, which sperm fertilizes which egg. So we talked about that 8.4 million number based on chromosome assortment. If you talk about both cells doing that, and then which sperm fertilizes the egg, you're looking at more than 70 trillion possibilities just on those numbers. Again, that doesn't even include crossing over. So this is why individuals don't have 2 identical offspring, other than identical twins. They don't have 1 kid and then 4 years later have a genetically identical kid, even with the same parents, whose chromosomes don't change in the meantime, because the odds are just ridiculously against it. Okay? And so all these sources of variation are very useful in terms of populations evolving over time. So mutations are one source of variation. However, we're getting other variation here with meiosis and crossing over. We can get new alleles, we can get different alleles, and some of these alleles might be more advantageous than others. This leads to differential reproductive success, so some individuals reproduce more than others, and then their alleles become more common. Natural selection is the process of the environment acting on this variation to increase or decrease various combinations of alleles. One example that's given a lot for human evolution is eye color, because for most of human history, we've generally only had brown eyes, very, very few other options. It wasn't until relatively recently a mutation evolved for blue eyes. And over time, the allele frequency changed. It went from nearly 100% of people with brown eyes to a significantly lower number. Even though brown eyes are still the most common eye color on Earth, now we have a lot more other colors, including the increasing number of blue alleles. That basically brings us to the end of our meiosis coverage. So if you have more questions on that, feel free to ask me about it, and you can go also online and check out our notes or whatever else you need. But thanks for watching, and I hope you got a lot out of it.